Hello everyone, welcome to my presentation today. My name is Bharat. I'm a PhD student at Northern Illinois University, where I work with Michael Papka and Maoyuan. I'm really excited to share our work on ChemoGraph, an interactive visualization system to enumerate and explore chemical space based on machine learning models behind the scenes. This work is a result of an amazing collaboration with researchers from University of Chicago and Argonne National Laboratory. Firstly, why is this an important problem? Well, let me try and convince you. As of March 2023, WHO reported that COVID-19 pandemic has killed nearly 6.8 million people worldwide. This is just one example, and there are many other diseases that can be solved with the help of small molecule therapies. This makes drug discovery an extremely important topic. With the help of AI and machine learning, it is only growing bigger. Yet only a tiny fraction of small molecules were explored by the scientific community. Recent research has also suggested that moving beyond standard samples from textbooks and lab stocks is extremely fruitful for finding drugs and novel materials. This motivated researchers for immediate and efficient automated methods of exploration and synthesis of novel compounds. But treating chemical space as a database listing of compounds without any structure is not useful. This is because such an enormous space is intractable for computational methods and they will ultimately fail. This motivated us to start envisioning the huge chemical space as a dynamic network. But how can people easily make sense of such networks? This is where a well-designed interactive visualization system helps. To be able to interactively enumerate the space and make sense of the enumerated space, we designed a visualization system. For the rest of my talk, I'll continue with a brief background and explain a few important terms and how we created a network out of chemical space, followed by the design considerations and a short demo of ChemoGraph. Then I'll discuss the initial case study we conducted with domain experts. And finally, I'll conclude my talk with comments on limitations and future work. Let me start with a term that we have been hearing for quite some time already, chemical space. So a chemical space is the design space consisting of every possible molecule. It is estimated to contain about 10 to the power of 60 compounds. And the building block of our work is scaffold. Now, what is a scaffold? Basically, a scaffold represents the core of a chemical compound. For example, given a chemical compound, we typically decompose its structure into rings, linkers, and side chains. Then, to capture the general description of global properties of the compound, linkers and rings are combined to form a scaffold which defines the core structure of the input compound. Consider this as a small collection of chemical compounds. Now, if we find the scaffolds of each of these molecules, we can easily identify that multiple compounds can have the same scaffold, that is, same core structure. This idea is the basis of our design because we can simply group the molecules based on the scaffolds. Firstly, this reduces the number of items to deal with, and second, it gives a better overview and organization of the space. Now we treat each scaffold as a node in the network. To understand how we make connections, let us look at various graph operations. The first operation is scaffold. It is simple and we kind of saw it already. Given a compound, a scaffold operation results in identifying the scaffold of the input compound, that is its core structure. Second operation is expand, the exact inverse of the scaffold operation. Here, given a scaffold, we try to identify the compound whose core structure is the given scaffold. But the catch here is we don't know how many compounds will have the same core structure. The third operation is called lower. Given an input scaffold, lower operation results in identifying the subscaffolds, that is the scaffolds whose structure is substructure of input scaffold. Finally, the last operation, that is upper. This is the exact inverse of lower operation. Upper tries to identify the super scaffolds of the given scaffold, that is, scaffolds whose structure includes the input scaffold as substructure. Scaffold and lower have straightforward algorithms to compute from an input compound. Popular software like RDKit supports these currently. But expand and upper require searching the entire space to find the results, hence sampling is used. As lower and upper are the only operations that operate between scaffolds, we use them to make connections between the nodes in our network. For detailed information on these operations and sampling methods used, please refer to this previous paper by our collaborators on scaffold embeddings. Coming to the domain goals for this design study, our collaborators mainly have two high-level goals. The first one is to design a platform that supports scaffold-based drug design. That means scaffolds should be treated as building blocks instead of chemical compounds and use the operations that we defined previously to navigate space. The second goal is that such a platform should be interactive and runs the state-of-the-art generative drug design models in the background to aid users in their exploration for finding novel compounds. To achieve these goals, we designed ChemoGraph. During the design, we looked at the most common alternatives available for visualizing different variants of network data, that is, node link diagrams, matrix visualizations, and list views. Matrix visualizations are not suitable for sparse matrices, and the resulting networks here are highly sparse as the ratio of the number of nodes to edges is close to 1. Moreover, matrices are not suitable for organizing more than 2 domains. Node link diagrams are the most used visualization types for networks, but their layouts are highly generic and suffer from visual clutter. They also do not suit for context slicing of nodes based on the inherent hierarchy present in our datasets. These are the main considerations which helped us in finalizing list views as our base layout, especially the support for context slicing. List views also offer better navigation of nodes based on the inherent hierarchy. Now let us see the illustration of the finalized design of ChemoGraph using list views. We represent chemical space as a hypergraph where nodes represent scaffolds. List view helps in arranging nodes into vertical lists where each list is defined by the number of rings in the corresponding scaffolds. The edges are identified based on lower and upper operations. Also, these operations connect nodes from adjacent lists only. This makes the hypergraph representation resemble a multipartite network. Here is a short demo explaining the components of ChemoGraph and how it can be used. Here is the visual interface of the ChemoGraph. Users start their exploration of the chemical space from the top control panel. There are two different ways to give inputs to the tool. First one is by importing files. ChemoGraph currently supports JSON and SMI file formats. Importing them will set up the initial view. Another way is to start by using command strings. We can either give a compound or prepend the compound with various operations and then hit submit. Here, I started with a lead compound identified from one of the high throughput virtual screening studies. This string representation is called SMILE string, one of the common ways to represent chemical compounds. To get an idea of some sample compounds in the scaffold class, we can run expand operation, give a sample size, and run the expand. Users can decompose a scaffold into subscaffolds by performing an operation called lower. Select the scaffold and then the operation. Similarly, users can create super scaffolds using upper operation. Users can perform these operations iteratively and can export the resulting networks. This helps the users to resume their exploration by loading a specific state. Now I am loading a previously saved state that I'll be using for the remainder of this demo. This process of starting from a lead compound and elaborating it into a series of local chemistries is useful in the generation of a structure-activity relationship series, where current practices involve by hand enumeration, and this is highly time-consuming. With this visual interface, users can do it easily while controlling the series generation depending on various attributes. Here we can see each scaffold is associated with two properties. The first one is log p value and the second one is SA score. These levels here represent the complexity of the scaffolds. Lower the number of rings, simpler the scaffolds. That means as the level increases, number of rings in the corresponding scaffolds also increase. This organization helps us in giving spatial structure to the scaffolds in the chemical space. Each level in the ChemoGraph can be independently sorted by a numerical attribute that is selected from the legend. Here log p value is selected, hence clicking on the level label toggles between different ways of reordering based on log p value. Also, changing the selected attribute to SA score updates all the levels correspondingly. Users can hover on a scaffold to see the directly related scaffolds. Selecting a scaffold highlights all the related scaffolds across all the levels. Changes in states, either for nodes or edges, can be clearly identified from the color highlights. This helps users to quickly identify both complex and simple scaffold classes related to the selected one. Zoom interaction is used to identify selected scaffold as substructure in the higher levels. In all the upper levels of selected scaffold, if the selected scaffold is present, zoom operation highlights the structure of the selected scaffold using a red color stroke. One of the important aspects of ChemoGraph is to provide the ability to take custom user-trained biophysical property prediction models as input and encode them on the scaffolds. By default, we have two common properties on all the nodes, but users can easily add their own models through the API, and the interface shows them here. So a user can select the model and encode it on all the nodes. So once the encoding is completed, the model can also be used as any other attributes like log p or SA score. For example, here I am selecting model 1 and all the levels are adjusted. Finally, once the exploration is completed, users can export either the smile strings along with the encoded properties into an SMI file or export the entire view as a checkpoint and resume their exploration in future. Coming to initial evaluation of ChemoGraph, we performed a qualitative user study with 8 domain experts in computational chemistry across various research laboratories. All the experts who participated in this study are well familiar with drug discovery tasks. Each participant, after watching the demonstration of interface, completed 3 tasks. Coming to the tasks, the first task requires analyzing biophysical property prediction models, and currently there are no tools to compare this with. The current approaches for tasks 2 and 3 require by hand enumeration of possible modifications to the input compound, and this is highly time consuming, whereas the participants achieved impressive completion times with our tool. After completing the tasks, participants filled in a questionnaire to provide their feedback on the tool. Overall, the experts rated ChemoGraph positively. During the post-study questionnaire, we asked experts about specific reasons that make ChemoGraph interesting to them. Here are a few responses that we got. When asked about new features that they would like to see implemented in the ChemoGraph, here are a few responses. Along with the suggestions from the experts, we also identified several limitations of ChemoGraph for future work. First is scalability. Though our goal is not to visualize the entire chemical space, we want our visual designs to be scalable enough to deal with bigger neighborhoods. Second is to provide a way to help users filter the space semantically. For example, we want to provide a way to remove nodes with unwanted chemical structures in the network. We ran an initial usability study to solicit feedback from domain experts, but we want to conduct a comprehensive user study comparing ChemoGraph aided tasks with traditional methods such as by hand enumerations. Finally, based on expert comments, we also want to investigate more useful ways of visualizing property models on the enumerated space. With this, I want to conclude my talk. Thank you for attending. For those who are interested in trying ChemoGraph, you can access our website at this URL. Thank you.